โอเคค่ะสวัสดีนะคะทุกท่านเลยนะคะผู้ที่ร่วมเข้ามาอบรมกับเราในวันนี้นะคะในหัวข้อการอบรมที่ชื่อว่าการใช้ฐานข้อมูลทดลองใช้ SciFinder-N นะคะซึ่งเป็นฐานข้อมูลที่ทางห้องสมุดนะคะเพิ่งเปิดทดลองใช้นะคะเมื่อวันที่1 ตุลาคมนี้เองนะคะเราจะทดลองใช้3 เดือนนะคะเดี๋ยวขออนุญาตแชร์หน้าจอเปลี่ยนหน้าจอแป๊บหนึ่งนะคะทุกท่านเห็นหน้าจอเว็บไซต์นะคะ
เห็นครับ
นะคะก่อนที่จะเริ่มเข้าสู่การอบรมนะคะซึ่งวันนี้เนี่ยเราก็ได้วิทยากรนะคะจากCAS มานะคะซึ่งเป็นผู้ให้บริการฐานข้อมูล SciFinder-N นะคะก่อนที่จะเข้าสู่ช่วงนั้นเนี่ยจะขออนุญาตแนะนำนะคะวิธีการเข้าใช้ฐานข้อมูลจากเว็บไซต์ของห้องสมุดก่อนนะคะเนื่องจากว่าฐานข้อมูลนี้นะคะทุกท่านจะต้องเข้าใช้ภายในเครือข่ายของมหาวิทยาลัยนะคะซึ่งขณะนี้ทางห้องสมุดได้เปิดให้แสดงตัวตนผ่านOpen-A10 เท่านั้นนะคะจะไม่ว่าเราจะใช้งานภายในเครื่องที่อยู่ภายในมหาวิทยาลัยหรือเครื่องที่อยู่นอกมหาวิทยาลัยนะคะจะต้องมีการแสดงตัวตนด้วย CL Open-A10 ทุกครั้งนะคะฐาน SciFinder นี่ก็เช่นเดียวกันนะคะวิธีการก็คือมาที่หน้าเว็บไซต์ห้องสมุดนะคะเดี๋ยวรบกวนน้องศิวพรคอยส่งลิงก์ให้กับผู้เข้าร่วมทางแชทนะคะอันนี้จะเป็นลิงก์เว็บไซต์ของห้องสมุดนะคะก่อนที่จะใช้งานทรัพยากรอิเล็กทรอนิกส์ทุกครั้งนะคะอยากจะให้ทุกท่านมาแสดงตัวตนผ่านตรงนี้ก่อนนะคะ CL Open-A10 หน้าเว็บนะคะซึ่งตรงเนี้ยนะคะไม่ว่าทุกท่านจะสืบค้นทรัพยากรผ่านกล่อง Zoo Discovery ตรงนี้นะคะซึ่งก็สามารถสืบค้นบทความ Book Chapter ฐานอิเล็กทรอนิกส์ต่างๆที่เป็นบทความหรือชื่อวารสารนะคะหรือว่าเป็นหนังสือผ่านกล่องนี้ได้นะคะหรือจะใช้ผ่านเมนู Database ก็แล้วแต่นะคะถ้าทุกท่านมาแสดงตัวตนผ่านตรงนี้นะคะก็จะอำนวยความสะดวกนะคะไม่ว่าจะเข้าใช้จากช่องทางไหนนะคะใส่บัวสี ID นะคะทำครั้งเดียวนะคะใช้งานได้8 ชั่วโมงโดยที่เรายังคงใช้เบราว์เซอร์เดิมนะคะพอแสดงตัวตนผ่านลิงก์นี้เรียบร้อยแล้วนะคะก็คลิกมาที่ Zoo Database นะคะหน้านี้เราปิดไปได้เลยนะคะก็จะเข้าสู่หน้าฐานข้อมูลออนไลน์นะคะซึ่งหน้านี้นะคะถ้าจะมาจากเว็บโดยตรงก็จะอยู่ที่เมนูนี้นะคะก็จะเป็นฐานที่เราจำแนกตามคณะสถาบันไว้ให้นะคะเรามีฐานข้อมูลที่บอกรับจำนวนมากนะคะเราก็เอามาจัดเป็นชุดให้สำหรับแต่ละคณะว่าแต่ละคณะใดนั้นมีฐานข้อมูลใดที่เหมาะสมนะคะในส่วนของคณะวิทยาศาสตร์นะคะซึ่งวันนี้เราจะเป็นฐานทางด้านเคมีนะคะคณะที่ใช้ประโยชน์ได้ก็จะเป็นวิทยาศาสตร์เคมีนะคะเภสัชนะคะวิศวกรรมเคมีนะคะอันนี้ก็จะเป็นฐานของคณะวิทยาศาสตร์ที่เราเอามาจัดชุดไว้ให้นะคะเป็นฐานวารสารฐานหนังสือแล้วก็ฐานรวมทรัพยากรนะคะซึ่งณตรงนี้นะคะจะยังไม่มีฐาน SciFinder-N นะคะเนื่องจากว่าเรายังไม่ได้บอกรับนะคะอันนี้เป็นฐานทดลองใช้อยู่นะคะถ้าต้องการทราบฐานข้อมูลทุกฐานนะคะสามารถคลิกที่ All Database ตรงนี้ได้นะคะตรงนี้ก็จะโชว์รายการฐานทุกฐานเรียงตามลำดับอักษรนะคะอยากทราบว่าฐานข้อมูลไหนนะคะมีเงื่อนไขหรือเป็นฐานเกี่ยวกับอะไรมีไกด์อะไรยังไงนะคะสามารถคลิกที่ด้านหน้าตรงนี้ได้นะคะตัว I ตรงนี้นะคะก็จะแสดงข้อมูลรายละเอียดเกี่ยวกับฐานนั้นๆนะคะวิธีการใช้หรือรวมทั้งคลิปการอบรมที่บริษัทมาอบรมให้เรานะคะเราก็จะเอามาแนบไว้ตรงนี้เช่นเดียวกันนะคะแล้วถ้าจะใช้งานก็คลิกที่ฐานได้เลยซึ่งตอนนี้เราแสดงตัวตนเรียบร้อยแล้วนะคะก็จะไม่ต้องใส่บัวสี ID แล้วนะคะทีนี้ในส่วนของฐาน SciFinder จะอยู่ตรงไหนจะอยู่ในส่วนของฐานทดลองใช้นะคะหรือว่าลิงก์จากเมนู Database ตรงนี้นะคะมาที่ลิงก์ฐานข้อมูลทดลองใช้นะคะแป๊บนึงนะคะเนี่ยไม่ไปก็คลิกตรงนี้ก็ได้ค่ะฐานข้อมูลทดลองใช้นะคะซึ่งตอนนี้เรามีฐานข้อมูลอยู่ฐานเดียวนะคะคือ SciFinder-N นะคะก็ ใช้งานได้ถึงวันที่31ธันวาคมนะคะต้องการทราบรายละเอียดก็คลิกที่ตัว i ด้านหน้านี้นะคะก็เป็นฐานทางด้านเคมีนะคะซึ่งก็จะแตกต่างจากฐานอื่นที่เป็นฐานบทความหรือฐานหนังสือนะคะตรงนี้จะให้พวกสูตรเคมีเราสามารถเขียนสูตรต่างๆได้นะคะแล้วก็วิธีการเข้าใช้งานนะคะจะต้องใช้ภายในเครือข่ายอย่างเมื่อกี้แนะนำไว้แล้วนะคะคือต้องแสดงตัวตนผ่าน CL Open ATM ทุกครั้งนะคะแล้วก็ผู้ใช้จะต้องมี Account ก่อนนะคะจะต้องมีการลงทะเบียนใช้งานสำหรับการใช้งานครั้งแรกนะคะก็สามารถมาดูรายละเอียดตรงนี้ได้ซึ่งเดี๋ยววิทยากรอาจจะแนะนำวิธีการด้วยหรือบางท่านอาจจะได้ลงทะเบียนไปเรียบร้อยแล้วนะคะหลังจากลงทะเบียนเรียบร้อยแล้วครั้งถัดไปนะคะก็สามารถมาใช้งานฐานข้อมูลภายในเครือข่ายได้เช่นเดียวกันนะคะเวลาที่ลงทะเบียนนะคะขอความกรุณาใช้อีเมลของมหาวิทยาลัยนะคะ @g หรือ @mswu นะคะวิธีการใช้งานก็ให้คลิกที่ฐานข้อมูลนะคะทุกท่านสามารถทำบุ๊กมาร์กไว้ได้เลยนะคะเนื่องจากว่าลิงก์นี้จะไม่ใช่ลิงก์สากลของฐานข้อมูลนะคะจะเป็นลิงก์ที่เราเอามาบริหารจัดการให้มีการเชื่อมต่อกับ Open ATM เรียบร้อยแล้วนะคะเพราะฉะนั้นบางท่านอาจจะไปใช้ลิงก์ตรงของฐานเลยเนี่ยจะไม่สามารถเข้าใช้งานได้นะคะต้องแสดงตัวตนก่อนโอเคตอนนี้น่าจะครบถ้วนแล้วนะคะตรงนี้ใครมีคำถามสงสัยสอบถามได้นะคะโอเคค่ะตอนนี้ก็ได้เวลาแล้วนะคะวันนี้เราก็ทางสำนักหอสมุดกลางนะคะก็ได้รับความกรุณานะคะจากอาจารย์อิทธิพลสังเวียนวงศ์นะคะภาควิชาเคมีคณะวิทยาศาสตร์นะคะที่กรุณามาเป็นผู้ดำเนินรายการแนะนำวิทยากรและช่วยเป็นล่ามแปลภาษาให้เราในวันนี้นะคะเนื่องจากวันนี้เรามีการอบรมวิทยากรของเราเป็นชาวต่างชาตินะคะน้องๆนิสิตหรือผู้เข้าร่วมท่านใดนะคะถ้าฟังแล้วมีคำถามนะคะสามารถสอบถามเป็นภาษาไทยได้เลยเดี๋ยวอาจารย์อิทธิพลจะช่วยแปลให้เรานะคะตอนนี้ก็ขออนุญาตส่งต่อตรงนี้ให้อาจารย์ได้เลยค่ะขอบคุณค่ะ
โอเคครับ thank you Sabri staff for setting up the trial session here and good afternoon all faculty members and grad students. I think most of us from chemistry department and I think we have some people from other departments also. So our training session today is we got speaker Pamela Un, the senior customer success specialist for Southeast Asia, Hong Kong, and Macau. Chemical abstract service, a division from ACS or American Chemical Society. So I think we can start the training session now. So please join me to welcome Pamela Un.
Thank you very much, Dr. Ittipol. Very good afternoon, everyone. So I'm very delighted to be here to share with you SciFinder-N. So I think some of you may have some previous experience using it, and it has some changes since the last time. So let's take a look at the platform. So I'm going to share my screen. And along the way, if you have any questions, please feel free to put it in the chat box, and I will check periodically to follow up with the questions. Okay. So first of all is to make sure you have a SciFinder-N account ready. So you can follow the instructions shared by the library earlier on to get your account registered. Go to the library website. Once you have your username and password, then you can log on. And this would be the page that you see, the landing page, okay? So in SciFinder-N, there are many types of searches that you can do, and you can find them on your left-hand side over here. The common ones would be reference searching, where you will look for journal articles, patent documents, all right? So it's like a literature search. Then you can also do a substance search, which is where you can put in a chemical name, a chemical structure, or even molecular formula, and you will find your required substances. From there, You can find literature, you can find properties, spectra of substances, okay? And then we also have reaction searching, where you can search for chemical transformations and find examples of how the reactions have been performed in the literature. What is slightly more recent that we have added in is the biosequence search capabilities. So this is for people working with protein sequences, nucleotide sequences. You may want to do a blast search similar to what you will find on the NCBI website. So you can now run blast search on SciFinder-N as well to find similar sequences. And then from the results, again, extend out to literature. So I wouldn't be covering this today. Okay, so, but if there's interest to learn more about it, please let the library know, and then we can set up a separate session to focus just on sequence searching, all right? So today's session, I will look at reference searching, substance searching, reaction searching, as well as retrosynthesis. So let's look at the first search type. So I always like to begin with reference search, because this is the one that is most relevant to everybody. As I've mentioned earlier, a reference search will help you find relevant literature. And this is the main search box where you would type in your query. You may notice that at the bottom here, there is a drop-down list. And over here, you will find additional ways to run a reference search, and you can actually combine multiple search terms into a single search. So I can say my favorite author, I can combine it with a certain journal or a certain publication year to find the required references, okay? And you have the Boolean operator options here to link up your multiple search terms, all right? But let's look at just reference searching by keyword. So I'm going to type in our first topic today. So let me type in mulberry. Oops, mulberry. Okay, and you can see as I'm typing, there is a drop-down list of word suggestions, which you can click to select for your query. If not, you can just continue typing. So I'm going to find information on the antioxidant properties of mulberry leaves, all right? So I just have my 3 keywords, type it in the box and run the search. So simple and easy. Okay, quick and easy. So this is the list of results that I get. You may see here that's quite a large number, over 100,000. And currently, the references are sorted by publication date. Okay. The next thing you may notice as I scroll down through the results, you see the blue hit highlighting that matches what we have just searched. And you may notice that it doesn't seem to contain all the required keywords, right? We see a lot of antioxidant leaves, but we don't see mulberry until the 4th answer. Mulberry shows up. And then again, we don't see all the required keywords. The reason for this is because when I put in a search containing 3 words, the system will try to find matches, references that match all 3 keywords, but it will also pull in the partial matches like the ones we are seeing here. So in some cases, partial matches are also useful because especially if your research area is very new, there's not a lot of work published. So the partial matches may be important for you as well. But in this case, you can see obviously we have a large answer set and we haven't even loaded everything. This is obviously not so ideal. One thing you can do is to change the sort options. Okay, so I'm gonna use our relevance ranking. Now the relevance ranking is very convenient and useful. because right now, after I applied the relevance ranking, it will pull up references that contain all the required keywords to the top. So you see now, antioxidants, mulberry leaves, antioxidant, right? So these are very relevant references using the relevance ranking. So the only drawback to that is you don't necessarily have the most recent publication. So right now we are looking at something from 2012, 2016. Okay, so to look at the more recent publications, we can use the publication year filter. So this is one way. I can always limit it to the last 2 years, hit apply. So in this way, I will still have the references sorted by relevance, but now I see the more recent publications. Okay, so this is one approach you can try to use the relevance ranking. Okay, use relevance ranking, and then you can use publication year filter to focus on more recent publications if that's what you're interested in. Another approach you can try is to make the search query, so this search query, make it more specific so that it doesn't pull back the partial matches. So one of the things you can consider using is Boolean operators. Okay, earlier on you saw the N or not, right? So you can use these 3 operators into your search query. So if these 3 words are required for me to be in the answer set, I would use Boolean operator N over here. Okay, so I insert N after each word. So this way the system will know that I require all 3 of these words in my result set. So you can use N, or, and not. So or is of course it will imply either you want mulberry leaves, or you want mulberry, or you want leaves. Okay, if you use not, 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 it would exclude. So if you say mulberry not leaves, so you're saying I don't want references that contain leaves. All right, so N would probably be the one that we will use most of the time to indicate all the required keywords. So if I rerun the search now, earlier on we had over 100,000 references, right? Now if I rerun, you can see out of the 100,000 plus results, only 563 contain all 3 of my keywords. And in fact, I should load this now because I have a more specific search query. Okay, so this will be my full answer set, 844 results that contains all my 3 required keywords. So you can see, even if I sort it by publication date again, right? I remove the relevance ranking. I sort it by publication date like before, but you will see the 3 required keywords now. Okay, so mulberry leaf, antioxidant, mulberry leaves, antioxidant. Okay, so this is the other way you can run your literature search, your topic search, to use Boolean operators, and then you can just sort them by publication date. Okay, let's take a look at one example of the results we see. So let's take a look at the first one. You have the title, the abstract, and then you also notice that there are substances. So to look at what are these substances, I can either click on the title directly, but what I like to do is to open this in a new tab. Okay, so I can continue to browse through the results here while this page loads up. So this is where you can see more information of each reference in your result. Okay, title, abstract, and all the blue hit highlighting. And then you have here the indexed information. So this particular reference, if you notice, is captured in CAS. That's why you see here CA+. It is also found in the Medline database. All right, so we also have Medline. So in SciFinder-N, you actually get to search 2 literature sources, the CAS reference database, and then for people who are working in, biochemistry, biomedical, where you're doing some cross-disciplinary type of work, where you need to search in Medline as well. Now you can just search in one place and search 2 literature sources. So the index information, let me explain a little bit more what this is. Concepts. Concepts is added in by CAS scientists. So we have a team of scientists who will actually read through the full text paper, and they would index or capture the important information that is described in the paper. So all these terms or keywords that you see here, they have been manually extracted out and input, and these keywords are important topics that are discussed in this paper. So you can see here that we index things like even plant gene. Okay, we index different biological entities that are important in the paper. We have things related to chemistry, flavonoids, and a lot of different information, as you can see here. Okay, so when you run the search at the top, it would also search in this section to help you find relevant references. So you can see Leaf has also the blue hit highlighting because it's also searching in this section. And Morris alba is the scientific name for mulberry. Okay, going down, because this reference is also captured in Medline, and Medline has their own way of indexing information. So we also have the Medline medical subject headings. So again, the keywords here would describe the type of research that is published in this reference, and the search query would also go into this section to find matching hits. Okay, and then finally we have substances. Substances would be important compounds that are involved in the research of this paper, and you notice that many of these substances, they don't actually have a structure because these are all biological entities. They could be enzymes, they could be proteins, receptors, etc. But we also index this and we consider them a substance. Okay, so maybe to, from the chemistry point of view, we may not really consider this as a compound or a substance, but in the CAS definition of a substance, a biological entity, biological molecules are also substances. And of course, then we also have the regular organic chemistry molecules that comes with structure. Okay. The other thing to note is, other than the name and the structure or the CAS registry number, we also capture the role information, as you can see here. Okay, so the role information is to give us a little bit more insights into how some of these substances, whether is it a biomolecule or an organic compound, how have they been utilized in the paper? Okay, and at any point, if you find any of these interesting, for example, this compound over here, you can click on the structure image. Okay, and this would give us the pop-up window where you can run a further search on a compound of interest. Okay, similarly, if it's a biomolecule, you could do the same thing. You just click on this. It will also come up with the pop-up window, and you can see, you can find reactions, literature, supplier, etc. Okay, so this is the information that you can find for each reference. Title, abstract, you'll have concepts. All right, let me just collapse it. You have concepts, you have Medline, medical subject headings, if it's also captured in Medline, and then you also have substances indexed. And after reviewing all this information, if this is interesting, you can click either on this link or this link here to get directed to the full text. So this will bring us to the publisher's website. And then, of course, at this point, depending on whether you have access, whether if the library subscribes to the paper, you'll be able to see the full text. Of course, in this case, this is open access, so I can actually load up the full text for you. Okay, so you can use one of these links to redirect you to the publisher's website. All right, now I'm going to go back. Now that you have seen the information, the indexed information that is available for you for each of the references in our answer set, let's take a look at the filter options, okay? So this is still quite a large number, right? So ideally, we want to be able to narrow it down further before we start looking at each reference in greater detail. So all the filter options are found on the left-hand side. Earlier on, we have already seen publication year, okay? So many other options to narrow down references. So a lot of these are also found in other databases, so I won't talk more about them, but I would highlight the ones that are special for SciFinder-N, okay? So the first one is the concept filter. Now you've already seen concepts, right? So just to remind you what is a concept, it's basically the keywords from here and from here, okay? From these two sections, all of these keywords, okay? These keywords, all right? DNA shuffling, droughts, they are considered a concept and they will be found under this filter option. So if I expand this, you can actually have an overview of all the concepts that have been indexed for the current answer set. So why is this useful? Well, first of all, if you look at top count, right, you will see that there are certain keywords that frequently occur in your answer set. So what does this mean? Probably it's an important concept. That's why it keeps recurring, right? So if you are just maybe learning about a research area, some of these concepts will give you some ideas what are important aspects of this research, okay? So as you can see, I'm scrolling down. You know, obviously we see antioxidant, mulberry, and leaves at the top because this is what we search. And then we see flavonoids also, you know, in the top 10. So this should tell you that flavonoids is an important type of compounds that you will find in the mulberry leaves, okay? And then if I scroll down further, you'll see things like anti-inflammatory agent, all right? So this is possibly some of its bioactivity, bio
นี้
effect.
เดี๋ยวต้องเอาไฟล์มาเปิดด้วยนะเพราะว่าครูอะเปิดใน
And then you
ไม่ได้
also have anti-diabetic agent, right? So maybe this is new information, or you didn't know that mulberry leaves can also be used to treat diabetes potentially. So I can select, okay, and then hit apply. So this will now focus the answer set on the literature that looked at mulberry leaves potentially for anti-diabetic purposes. So you can see now the results, 53 results, we see anti-diabetic in the title. In this case, again, we see anti-diabetic compounds. Okay, here I think it's the hypoglycemic. It's also indicative of related to diabetes. Okay, and then let's see what we have here. All right, so if you don't find the keyword in the title or the abstract, remember there's the concepts to double check, all right? So now you can see how this is also one way to help you narrow down answers to a specific topic of interest, all right? Let me open this up again. And so remove this. The other thing you can do is actually search for keywords of interest, okay? So maybe I'm interested to find out what are some ways that I can do the extraction on mulberry leaves, right, to get the active components. So I can just search for the word extraction. Then you can see here terms related to extraction pulled back for us: microwave extraction, ultrasonic, supercritical, or I can search for words like chromatography if I'm trying to find or understand what are some chromatographic techniques to analyze the plant extracts. Okay, so I search chromatography, and again, you see many of the terms related to chromatography pulled back for us, and I can even select multiple concepts. Hit apply. Okay, so besides looking at top occurring concepts, you can search for concepts of interest. All right. Then just to show you how this concept filter is working. So like I've mentioned, we may or may not see the word chromatography appear in the title or the abstract. The important part to check, okay, is, all right, let me open this up. So in this case, it did say LCMS MS, so it's mentioned chromatography, but the concept filter is really looking at this section over here. Okay, so it has found liquid chromatography mass spec indexed in the concept section. Therefore, when we apply this concept, okay, one of the ones here, it finds us this set of results. Okay, so this is the concept filter. Okay, let me clear this. So this is a useful one to explore. I'll definitely encourage you to take a look at this. The other useful filter, it's definitely going to be the search within results. So this is where you can put in your own choice of keywords, right? So concepts is limited to the standardized terminology used by the database. Search within results, you can put in your own choice of keywords, because maybe sometimes something that you're looking for has no standardized terminology to use. So I can just put in my own keywords. For example, I now want to see if there's any paper that look at the activity against the acetyl cholinesterase, okay, acetylcholinesterase. So I'm going to type in, so this is I guess it's some kind of a enzyme, a biological entity. So I'm going to type in into the box here. Let me zoom in. Okay, acetylcholinesterase. All right, so let's see if any papers reported on the activity against this biological target. Okay, we found 6 results. And the search within results will search title, abstract, concepts, right? Just like the initial search over here. And you may notice, and you will see additional blue hit highlighting. Okay, so this is how you can, so like I say, if it's not in the title abstract, just take a look at the concepts. Okay, over here again, we see it appearing. All right, so search within results is another useful filter option to use to find relevant literature. Once you have run your search, you have filtered it, and you got a nice set of results. Maybe you are using it, you're citing it for your own papers or your own research. You can download this 6 results, okay, in one of these formats, and the citation format will be the one to use if you are going to use EndNote Mendeley. All right, so save it as a citation format, and then you can open the file in your reference management software. You can also choose to save the search and set up an alert. So this is the button to use. You can choose to save just the search, so that it will remember the filters you have used. Then you can rerun it easily in the future. Or you can choose to save the search together with the answers. Okay. And this is where you can set the alert frequency. So let's give it a name. Mulberry leaves antioxidant. Okay. And then remember to hit the save button here. So whatever you have saved will go into this folder. Okay. So let me open this up. And we can take a look. All right, so this is what we have just saved. So you can see it remembers the search query as well as the filters that I've used. I can come in here to rerun the search, and I can even view the results if I have chose to save the results as well. So in this case, this is something that I saved previously. I didn't save the results, so there's only the rerun search button. Okay, now you only need to save the search, like I say, if you want to set up an alert. All right, you can change your alert settings here, or if you want the system to remember the filters that you have used, right? If this is not required, then you don't actually have to manually save, because we automatically save all the searches that we run in the box, in the initial query box. Okay, so let me show it to you. There's the search history button on the top top right hand corner, this clock icon. And then this is where you can see see all the search history. Okay, let's wait for it to load. Let's try it here. Yeah, okay, it's loaded. So you can see these are the two searches that I've run this morning, automatically saved for us. And in fact, I have the calendar here, which will allow you to recall searches that you have run months and months ago. So a little tip, if I want to see the searches run on the 18th of May, you can see that the cursor is on the start date box. So instead of clicking 18th of May right away, click on the end date box first, and then select the date that you are interested in. So in this way, it will show you the searches performed on the May 18 directly, okay? Like what you see here. Right, so I've only run one search. Okay, so the calendar, you can use the calendar to recall searches that you have run a long time ago. Okay, now going back to our reference search. So we've looked at exporting results, saving results, setting up alerts. The other thing that would be interesting is to look at cited references, the citation map. So let me find something, okay, like this. You see that it has cited 2 times, right, by other people, and this reference obviously cites some literature as well. So we can open this up in the citation map, and then this will allow us to see both the cited and citing references. They will be listed in these 2 tabs, okay? And you can click on the tabs to read the abstract, and of course go into the title to look at concepts and substances. All right. You can also do an expand citation. So these are relatively new, so that's why it's been grayed out. Let me show it to you on this direction. Okay, so let's find one that has options to expand. Okay, so you can see here, you could theoretically, in theory, expand citations in both directions if there are citations to be expanded. Okay, so just hover over the circle and see if you can click on this button. Okay, and there is also filter option within citation map. So, you know, there could be a long list of references to review, right, under these 2 tabs. So instead of clicking on every single one to look at the abstract, I can first use the concept filter and pick what is the topic that I'm really interested in. So maybe, for example, we were interested in acetyl choline esterase. I can pick this as a concept and just focus in on the references that also contain acetyl choline esterase as a important aspect of that paper. Okay, so this is the filter options found in citation map. Okay, so this pretty much covers reference searching. So just to do a quick summary of what we have talked about, a few key things to remember. Use Boolean operators in your search query if you want more specific results. So this would be things like and, or, not. There's another one that I didn't demonstrate, but it's also available to use. So for example, if you want mulberry and leaves to occur together, okay, you can also use things like this, double quotation. Okay, so this is a lot more specific, right? This requires mulberry and leaves to be right next to each other. So I didn't use this for this example. I didn't add the double quotation because of sometimes people write it this way, right? Mulberry, Morris, Elba, L leaves. So you can see it's split apart. So if I have searched this way, I won't be able to find this paper. So just be mindful, all right, that if you're using the double quotation, the two words need to be right next to each other. So in this case, what I think is a better approach is to just use the and operator. So and or not, double quotation, if you want even more precise results. And then make use of the sort options. Relevance ranking is always convenient to use because it pulls back the best matches right to the top. Okay, so you don't have to go through the entire answer set to find the relevant references. And then in terms of filter options, all on the left-hand side, the ones that I would highly encourage you to explore would be the concept filter, as well as the search within results. Okay, and then remember that you can always save your search. You can set up alerts. So this will help you to rerun searches easily and also help you to keep updated on latest additions into the database. Okay, so this is for reference searching. Now I'm going to move on to another type of search on SciFinder-N. Just take a quick pause to see if there are any questions. Okay, no questions. So remember, if you have any questions, please feel free to put them in the chat box, okay? All right, let's look at another type of search on SciFinder-N, which is substance searching. So like I said earlier, you could find substance by its chemical name. You can put in the structure, molecular formula, and in fact, if you click on the drop-down arrow, there is so many more ways to find substances by a certain chemical property, physical property, biological property, or even NMR chemical shifts, okay? And like before, you can click on the plus sign to combine multiple search terms in a single search. So let's look at just a substance name search. So I'm going to type in our substance example. So usually for substance searching, I like to type it slowly because I like to see if it will appear in the drop-down list. This can help me to double check spelling. It can help me to double check that I have the right format. Because sometimes you can see there's a space, sometimes it's a dash. If you're not sure, just type it slowly and see if it appears. Okay, so I actually find the compound that I'm interested in. This one over here. And let's run the search. Okay, I'm going to close some of the previous tabs. All right, so this is the compound that matches our substance name. you can see that there are 35 associated literature for this compound and 1 supplier. I can click on the CAS registry number or open this in a new tab to look at more information like properties, spectra, etc. Okay, so if I scroll down, you will see some experimental properties. All right, so this is relatively new compound, or I should say not so much work has been published on it, just 35 results, right? So we just have 1 biological property listed here. And then we have 1 carbon-13 spectra. So for different compound, there's going to be different amounts of information available. So just click into the 2 tabs here just to see whether the property or the spectra you are looking for is available in our database. Okay, now if I scroll down further, this even has a bioactivity indicator. So although only 35 results, but there are actually 9 of the 35 that looks at this compound as a potential anti-tumor agent. Okay, so you can click on this this link here to look at the literature that explores this bioactivity. Okay. And then, of course, if I scroll up, I can go to references. I can go to supplier information. Now, this is searching the compound by name. Now, we're going to do the same compound, same example search, but instead of name, I'm going to put in the chemical structure. Okay. So one quick and easy way to add a structure into the editor, if it's already available in SciFinder, is to click on the structure image and then click edit structure. All right, so this transfers it directly into our editor. I'll talk about the drawing tool later on. First, let's just run the search on this structure, okay? So click OK. All right, it's uploaded into the search box, and I'm going to run the search. Okay, now we've run the same search for this compound, but by structure, and we get a lot more results. So under Estron, Estron would be compounds that match the same structure query as what we have drawn here. It has found 2 results, but I'm only seeing 1 here because I have a stereochemistry filter applied. So the other compound here, the second compound here, probably is a stereoisomer or maybe no stereochemistry. If you want to see what is the second compound, we can always remove the filter. Okay, so if I zoom in, you will see that it has the same structure, but it doesn't have the double bond geometry specified in this molecule, in this compound. So it has a different CAS registry number, is considered a stereoisomer. So under the Estron, you can find things like stereoisomers, you can find your isotope labeled compounds, you can find salt forms, mixtures. All right, they'll be all under the Estron as long as they contain the initial structure query. The second answer set is the more interesting one because it allows you to find not only your structure query compound, but also related or structurally similar substances. Okay, so right now I have my substances sorted by number of references. Let me switch it back to relevance. All right, so you can see this is our structure query, all right, coanine. And then you have here the second compound that is also found, but under substructure. If you look closely, the difference is that this has an additional alcohol, right, at this position. Okay, but this is structurally very similar, just with this additional substituent. Same thing, this has the modification happening at this part of the molecule. Okay, so the substructure search will allow you to find such compounds. that contains this structure query as we have drawn, but with additional substituents. Okay, so this is a substructure search. Now, the last one is similarity search. It's also interesting. It also allows you to find structurally similar compounds. And there is an overlap between these two, but this has some unique compounds as well. So if you compare, this is again choanine, right? It's our structure query compound. If you compare to this, it has a score of 100, which means it's, you know, very closely matching our structure query, but it has no highlighting in blue. Okay, the reason for this is if you compare the two structures, you will see that this doesn't contain the same basic scaffold drawn in our query, right? So I think it's because on this chain here, this is much shorter than what is originally found in the structure query. So this is something that you won't find in the substructure, but it will be found in similarity. Okay, so it's also similar, but just doesn't have this structure query present anywhere. Okay, now if I scroll down, you'll see many of such substances. And then the ones with the blue hit highlighting will be the ones that you also see in substructure. So this is the difference between these three answer sets. Okay, substructure will always require the structure query to be present in the results, but it can have additional substituents. Similarity uses an algorithm to score compounds, so it doesn't actually require the structure query to be found in the results from similarity. Okay. Now, so we have here, 448 results, quite a lot of compounds. You can make use of filter options available to help you prioritize what are important compounds possibly for your research purposes. So one of the filters you can consider using is the reference role filter. So this tells us how each of these compounds have been used in the literature. Okay, so I can open this up. So depending on what is it that you are focusing on, whether are you interested to look at pharmacological activity, whether you are interested to look at, whether it's being isolated from a natural product, or if your compounds are being used as catalyst, or if you're trying to find analytical information, we have, let me see, analyte filter, analyte reference role. Okay, so a lot of interesting reference role information that you can use here. So maybe I will select natural product occurrence. Okay, so I'm interested in the compounds that have been isolated from natural products. So I have here 179 results because I've limited it to a score of 90 and above. If you want to consider more compounds, then you can select the lower score options. Okay, so 179 results, all of this here, they have been isolated from natural products. So we might be interested to see what sort of plants, which papers have published all this information. We can use the reference button at the top and just cross over into the literature. Okay, so you can see here the 1,654 results, and then this would have our compounds from, actually let me open this in the new tab. Okay, so I could, so this set of results would mention these 179 compounds. We can do some filtering. Again, I can open up the substance role and focus it in on natural product occurrence. So it brings it down to only 200 references. Okay, now if I use this as an example, let me open this up. You will find under substances, so it should be this compound that was from this list here, okay? And you can see it has the natural product occurrence role assigned to it. Okay, so so this is how you can cross over from substances back into literature. And then of course you can use any of these concept filters that we've talked about, or even search within results to find even more relevant literature. Okay. All right. So this is searching substances by structure. Now, for people working with inorganic compounds, of course, you can use substance name search. You can also use structure search, but probably you would find using a molecular formula most convenient. So let's look at an example using molecular formula. So I want to find molybdenum tungsten oxide. Okay, so instead of spelling the whole thing out to search it by its chemical name, I can put in the molecular formula. So molybdenum, MO, tungsten, W, oxide, which will be oxygen, O, and then I know that it's 3, I think. Okay, now I'm going to run the search. Okay, actually, I think it should be 4. So 3, there's also something exists as 3, but let's look at the ones that has 4. Okay, so this is one way to search molybdenum tungsten oxide. And then you have here tungstate 1 minus. So I think the tungsten has a different oxidation state. But these would be 2 compounds that matches what we have indicated in our molecular formula search, right? Where there's 1 molybdenum, 1 tungsten, and 4 oxygen. Now they are displayed to us in this table format because these are compounds where we don't actually have structural information, or these are crystal 3D lattices. So it's not possible to display its connectivity using like just a single molecular structure. Okay, so these are called tabular inorganic substances. So what we do is we just index the individual components and the ratio information. All right, so this is one type of molecular formula search that you can do. Now, if you want to do something even more advanced, okay, or something broader, where you are just interested to find compounds that contain molybdenum, tungsten, and oxygen, but you don't have a specific ratio, right? It doesn't have to be MOWO4, it can be O3, or it can be any other ratio. There is another type of molecular formula search you can do. So let me modify this. Okay, the same thing, I have molybdenum. I put in the period symbol, okay, so full stop. I put in tungsten. And I put in oxygen, all right? But I don't have to say how many molybdenum, how many tungsten, how many oxygen. Not necessary. Just put in your elements of interest and run the search. So you can see this is a much broader search, and it pulls back everything in our database that contains these 3 elements, either in unknown ratio or in any ratio that has been reported. Okay, so you can understand why there are these, compounds where you can't really search them with the standard molecular formula because the ratio is actually a range, or the ratio could be, a decimal place. So there are these more complex type of materials, right? That it's hard to use the normal molecular formula way of searching to find it. So with SciFinder-N, you have a very easy and convenient way. Just put in the elements that you require, okay? And then you can see, what are all the different substances of molybdenum, tungsten oxide that have been reported in the literature. And just like before, you can always just cross everything over into the literature and find out what type of work has been done on it, what has it been reported for. Okay, so you can see your solar cells. You can take a quick peek into our concepts, and this will tell you at the moment what sort of work have people done using those compounds. Okay. So this is substance searching. So again, a quick overview of what we have covered. Substance searching by name, which is always the easiest, but remember that it works well only if your compound name is short and simple, right? Once the name gets quite lengthy, it's probably easier to search it by structure or even by molecular formula. And for molecular formula, you have the standard approach where you put in the exact components and its ratio, or you can use the dot disconnect formula where we just put in the elements of interest, right? So let me just repeat it again. Elements of interest followed by a full stop. And then we don't have to say how many oxygen, how many tungsten, how many molybdenum. All right. And then for each substance, you can always find information like properties, spectra, or you can cross over into the literature. And then remember to use either the substance role or the reference role, okay, or the reference role to help you choose or prioritize the relevant compounds for your research, okay? All right, let me take a quick look. Okay, so I've seen questions. Let's address it. Can we combine Boolean operators in a search? For example, if I want to search about in either leaves or roots. Yes. Okay. So the answer is yes. We can do a combination of N or in the same query. So using the mulberry example, right? So it can look something like this. Mulberry and leaves or root and antioxidant. Okay. So for the Boolean operators, the OR operator would always take precedence over the AND, which means in something like this where there is a combination, it will actually group these two together first. Okay, so it will look for leaves or root, and then it will combine it with mulberry and antioxidant. So you could actually just search it directly this way. But what I sometimes like to do, because I sometimes, I just want to make sure that the operators are applied in the right order that I need it, you can add in parentheses. So this makes it a little bit clearer which words should be combined with which operator. So you could also put in this way, so it's very clear, leaves or root, and mulberry and antioxidant. Okay, and in some cases it does actually matter. It depends where you put your and your or. Okay, so So yeah, so either use parenthesis or just remember that the or is always highest priority over the end, and end is higher priority than not. So not is the lowest priority. Okay. And then the other question. Okay. So for charged species, so if you search molecular formula, it will pull back. You can't use the molecular formula to search specifically a charged species, but it will pull back everything, whether it's charged or non-charged, it will pull it back. Okay, I don't have a good example on hand to demonstrate it. Let me have a think. So even if I use the molecular, I think it was this one, right? I got two results. So this is not really charged species, but you can see how it pulls back where tungstate, where there's a 1 minus. I don't know whether is it on the tungsten or the oxygen, but this is definitely has an oxidation state 1 minus versus this, which is so-called neutral. Okay, so I just searched this single molecular formula. It will pull back the charged species, the free radicals, the ions, etc. And in this case, it's only tabular inorganic. Sometimes you can go into the substance class, okay, and then you can find there's a substance class for radical ions. I'm assuming that's the kind of compounds you want. Okay, so in summary, right, whether you're searching by molecular formula or by structure even, okay, it will pull back charged species, neutral species, everything together. Then you have to kind of eyeball it a little bit to find the exact species that you want. All right. If you have an actual example, then maybe it's easier to explain. Okay. The other thing that I will quickly mention about maybe not exactly charged species, but with metals of different oxidation states. Okay. So let me just let me draw a simple let's hope I can find something. All right, so let me just draw a simple, like a metal ligand type of complex. Okay. So I'm just keeping it general so that hopefully I get some hits. Okay, so some people will say, well, I only want iron 2 or I only want iron 3. How do I search such species, right? So you can use the this plus or minus sign to indicate the oxidation state. So let's put it as 2+, all right? And then I'm going to run the search. So I hope it has something because I've kind of made this up on the spot. So let's hope we get something. Okay, let me go to substructure. All right, and let me just maybe limit it to so earlier on I was mentioning, right? So there's a substance class radical ion, but I'm going to limit it to coordination compounds. Okay, let's just use whatever I have. Maybe let's do single component to keep it simple. Okay, so you see here that this is iron 2+, this is iron 3+, right? So although in my structure, I did indicate that I want it to be iron 2+. So it finds me all the oxidation states anyway. But there's one way to make it more specific. So this is only if you're using a structure, searching by structure. There is the option to do a structure precision. So in this way, you can then say, I want to focus on conventional results, okay? And this should find you just the 2+. All right, so you can see now, because I select conventional results, the answers that I'm getting now is quite small, but now the ion center is now, they're all 2+ now, okay? So the 3+ gets pulled back under tautomers and Zwitter ions. So I know it's not the conventional definition of a tautomer or a Zwitter ion for the 3+, but we kind of lump it under this category so that it's convenient for people searching metals with they're interested in multiple oxidation states. They don't have to search each one individually. Okay, so it pulls back all the oxidation states. But for people who want to be more specific, then you can use the structure precision analysis and then select conventional results. Okay, now similarly for the tungsten molybdenum oxide example, you can also use that search using molecular formula. All right, so let me remove it. And since we are talking about searching inorganics by structure, let's do it for that. So you need to open up the periodic table, molybdenum, tungsten, oxygen. Okay, so it is also possible to find those tabular compounds using this method, but this method is of course broader because now you could possibly also find metal ligand type of compounds, right? Because now it's a structure search, and once you open it up as substructure, then you can have ligands substituting at these metal centers or even on the oxygen. But you will find those tabular compounds as one of the results as well. So let's just run the search and you can see what I mean. Okay, so for the estron, it's probably okay. So that's where you get all your the tabular inorganics, like when we did the molecular formula search. But you can see this is over 4,000 results, right? So let me show you why there are more than. Okay, so there's more than 100+ results. When we search by molecular formula, we only had 139, I think. Okay, do I still have it open? No, all right. But you search it by structure, there's over 4,000 results because now you're allowing a fourth component to be added in, such as these answers. Okay, so that's the difference. If you use number of components, you can limit it to 3, which is the 3 that we have searched. Okay. All right, so this would be similar to when we did the molecular formula search. But the structure search will allow you to search additional components. Okay, that's one difference. And then if we go into the substructure, you can see the number is even bigger because now you would have let me just show you. You have this sort of coordination compounds being pulled back as well. So you can see it's coordinated to other stuff. Okay, so I don't know if this is considered like a charge compound plus. Okay, I guess not. No, plus 2 minus, there's one more minus here, so maybe possibly. Okay, so yeah, so inorganic compounds, you can also do structure search, but just be aware that it gets even broader. Okay. Okay. All right, so hopefully that kind of answers your question. Then I'll move on to reaction searching. Okay, so reaction searching. Now, reaction searching, there is the option to do a reaction search by text. So you can put in your product or your target molecule in as a substance name and find reactions, or you can put in a catalyst or a reactant. But I think most people who do reaction searching would probably search it by structure, right? The other way is, of course, if you find something like this, let's see, if you find a compound, if you can find a compound in our database, you can always click on the reactions button. All right, so this is a quick way to find reactions without drawing anything. Okay, so over here, right, I click on reactions, it opens up to reaction schemes. All right, then that is if you search for a substance first and then find reactions. Now, if you are starting for reactions search from scratch, like I say, most probably you'll be drawing out your reaction scheme or your target molecule instead. So let's take a look at that, okay? So we have the drawing editor here. And if you have already drawn your structures in ChemDraw, usually people use ChemDraw, you can actually export it into, export the ChemDraw structures as a mol file, and you can import it into SciFinder-N, okay? Let me see if I have some examples. Should have, I think. No, I don't have training materials. Structures, yeah, okay. So this is just an example. So you can upload it. Let me open up my ChemDraw. Okay, so this is one way. The other way is you can also convert your ChemDraw structures into a SMILES string. So if I select it, go to Edit, Copy as SMILES, then go into CASDraw, paste the SMILES string here. Okay, and then it converts it into the structure. All right. Earlier on, I was saying about saving it as a mol file, right? Let me just make this a little bit more complex. So if you want to save this as a mol file, go to Save As, and then select this, okay? Don't select the V3000, select this format. And then this is the one that I've imported in just now. So export as mol file, convert into SMILES string. The last one would be if you have ChemDraw 18 and above. You can see once I select the molecule, I do have some add-ins, and the add-ins have the SciFinder-N option, which you can launch the search directly from your ChemDraw structure. Okay, you can select whichever is the search that you want to draw. The only thing to take note of is, especially for your university, because you're accessing SciFinder-N via the Open Athens proxy. So remember to add in the correct URL with the Open Athens proxy attached to it, or else it will open up the public domain SciFinder-N, which is a different URL. Okay, so just paste it in, and then you can use the API, the add-ins in ChemDraw. So a few options to choose from. If not, just draw the structures in SciFinder-N from scratch. Okay. So let's take a look at the drawing editor. So I've added in already the benzene ring. So the commonly found ring systems, used ring systems are found here. Let me continue building on this. I'll attach another, okay, another benzene ring, and I'm going to select 5-membered ring. Now to convert a carbon to oxygen, you can select oxygen here and then click to replace, or just like in ChemDraw, you can remain on carbon. Just bring your pencil to the node that you want to change and press O on the keyboard. It can also replace it with oxygen. Okay, and then I have OH. Then if I want to connect OH to this position here, I can click to add the carbon bond first and then replace it. Okay, so this will be my starting material. So let me just push it one side. I can do a copy and paste. All right, and I'm going to do a bromination reaction. So I'm going to attach bromine here. So if I have bromine selected, I can't just click, all right, because it just replaces the carbon with bromine. So let me undo this. So what I need to do is left click on the mouse, drag out the bond. Okay, you can see I'm still holding on to my mouse, and once I release, the bromine is attached. All right, so I think it's pretty intuitive. You will figure out it's slightly different from how you draw bonds on CamDraw, but I think you will get used to it after a while. So the next thing I would usually do is to add in the reaction arrow here. All right, so this recognizes that this is the reactant, this is the product. So of course, when you do a reaction search, you don't always need the full reaction. You can always just put in the target molecule. You can just put in a half reaction. You can run the search, or you can just put in the reactant and it finds you reactions involving this reactant. So the full reaction is of course going to be more specific. Okay, so let's run this. Okay, it's uploaded into the search box. Click the search button. All right. So right now I have my answers sorted group by document. So under as drawn, I just have one reference, and then this is the reaction scheme and reaction conditions. If I go to my substructure, so substructure is similar to the explanation that I've shared earlier on for substance searching. It finds reactions that has the basic structure query, but then it has, you know, additional substituents, all right? Or like in this case, okay, so this is still a bromination reaction. So in this case, this is not, this bromine is already here. So this is not so useful, but something like this would be useful. Now, if you don't want to see answers like this, right, where the reaction is actually happening on another part of the molecule, I can go back to my reaction query and indicate where the reaction site should be. So I can say over here. So in this way, I would make sure that whatever reactions I get, the bond formation or the bond breaking is happening at this bond here. Okay, so if I rerun the search, we should get fewer hits here. Let's take a look. Okay, only 5 now. And the earlier reaction we saw where, you know, it's actually putting something on the phenol group is removed. Okay, so you can see these are adding the bromine. This is also adding the bromine onto the molecule. Okay, so this is viewing by document, but I would typically view them by scheme. So this is where I can see individual reaction schemes. Okay, so this is the one that's matching exactly what we searched. This is a substructure, and then these are different substructure examples. All right, and then they could be from the same reference. Like you can see, this is one scheme from this paper. This is a second scheme from the same paper. So some people, they don't want to see the same reference, you know, appearing again and again. So they choose the group by document. But if you want to see every specific scheme, then you choose group by scheme. And sometimes there could be more than one reaction for each scheme, which means that more than one papers have performed that exact transformation. And then they may use a different reaction condition. So we'll group them together so that you can just look at all the different possible reaction conditions. All right. So my example here, everything, there's only one example, one reaction. Then the other important useful information for reaction searching is, of course, the protocols. So you can see this has the link here. And this just means that you can view the protocol in SciFinder-N, okay? So it will be, yeah, the details here. And this is, of course, taken from this paper. Okay. Now, if it doesn't come with the experimental protocols, then like in this case, then you'll have to go to the full text to get the detailed synthesis. There is the experimental protocol filter, which will tell you which reactions come with synthesis that you can view in SciFinder-N. Okay, so I'm not looking at this because this sometimes can be a little bit different, but I mean, if when you are desperate, maybe you can take a look, all right? Because this would, you can see it's, you know, it can be quite different from your initial starting structure. Of course, you can make it more similar. All right, but still it's compared to what you see in the substructure. This is only matching very limited space of, you know, what we have drawn here. Okay, so I don't normally use this for reaction searching. Okay. Now, so if you were planning a synthesis, right? So you found a way to synthesize this. It may be that you need to also synthesize this because like there's only 3 suppliers. Maybe it's expensive or it takes a long time to get delivered. So you may want to synthesize this yourself instead. So to find reactions on how to synthesize the starting material, you can click on the structure image and you can say synthesize. Okay, so this would then show you the reactions to synthesize this. Okay, now if I cross over. All right, so now you can see this is now the target molecule. And then this is an example where we have more than one reaction per scheme. So there are 6 examples where this gets transformed into this. And then these are the different papers with the different reaction conditions. Okay, and then again, you can use experimental protocols to find the ones that have the synthesis available in SciFinder-N for you to look at. But we now have the retrosynthetic analysis function, so you don't really have to do it step by step to plan your synthesis. You could just submit this molecule into our retrosynthesis planner, and the system will go into our database and pull back all the different pathways that will allow you to get to this target molecule from simpler building blocks. Okay. So to access the retrosynthesis function, you have seen already when I click on this, there is the pop-up window. I can say start retrosynthetic analysis. That's one way. In the reaction scheme that we have submitted, it will also, you can also do a start retrosynthetic analysis, but it will only take the structure of the product. Okay. If I go back to the home page, you can see there is also a retrosynthesis function button, right? So let's use the one here. I'm going to put in the same molecule that we were looking at earlier on, 1804111-11-4. Okay. All right, and then just start retrosynthetic analysis. So it brings us to this plan options page, and then we can do some customized settings. You can indicate how long you want the retrosynthesis to be. The default is 3 steps, and I typically leave it as 3 because if the system is able to break it down further, it will expand it for you anyway. So I just leave it as 3. if you set it to 4, it just takes a longer computational time. All right, so it's up to you to decide what is the best balance. All right. The rules here is referring to predicted reactions. So even for something that is already known in literature, like this compound that already has known synthesis, when you submit it to a retrosynthetic analysis, it will suggest new ways to make it that has not been reported in the literature before. Okay, so this is the predicted reactions, and you can choose to see predictions that are based on common chemistry. So common chemistry would be chemistry that has been published and reported many times, like your coupling reactions, reduction, your acid coupling reactions. Okay, then there are the slightly less common ones and then the rare ones. So you can choose again which set of rules do you want. I always just leave it as common. Again, including uncommon and rare would increase the computational time. So I would only use this if there is some very unique scaffold that probably needs some very specific chemistry to get to the scaffold, then I would include these. Okay, if it's just normal looking motifs, I just leave it as common. And this is where you can set the starting material cost. The default, I believe, is $1,000. So for you, if you're using it for the first time, you will see $1,000. So you can set it to whatever is a comfortable, I guess, budget for you, right, in terms of purchasing starting materials. So I just set it as $25 US dollars per mole. You can play around with this. The higher you set the starting material cost, the more complex starting materials it will suggest, right? Because it will just try to meet the highest budget that you have indicated. Okay. So once you've set all this, just click on create. So what happens next is if there are any experimental pathways, it will usually be computed fairly quickly. So you can see already one pathway shown to us. Okay, but you can also see that it is still generating. The predicted results will take a little bit more time. So what I'll normally do is just leave this to run in the background, and then come back again to check later on when everything is done before I start reviewing. Okay, so I will go to my history to pull back. Oh, it's already done. So it's pretty fast. Okay, so sometimes you'll still see that it's running. All right, so this is generating. So since it's done, let's open this up. And then it's telling us that the predicted steps are ready. So I'll say, yeah, okay, let's view them now. Or maybe let's turn it off first. Okay, let's just view. Okay, so this is just one of the pathways that it has suggested for us. Okay, so it's a retrosynthesis, right? This is the target molecule. It breaks it down and it starts with this as the building block, which matches our our starting material cost limit. Okay, so as I said, this is only just one pathway. There are a few things you can do to look at alternatives. One of it is the scoring profile over here. So this pathway is shown to us first based on this scoring profile. And this is the default scoring profile that you will see. And then you can make changes to it depending on which criteria is most important for you. So for example, I may look at this and say, well, the yield looks a little bit low. Is there any other pathway, any other retrosynthesis pathway that would give me a better yield than this? So I can go down to my scoring profile and I can push this up to high. So this is saying that yield is the most important criteria for me. So please help me find something with a better yield. You can also push this down just to make a greater contrast between yield and the other criteria. But let's just try with this first and then hit apply. So it will go into the system, look at all the different options and come up with something. So if you look at the pathway, it has already changed. Okay, so it's giving us a different starting material to start with. And if you look at the new pathway suggested, it has definitely a higher yield than before, 46%. But of course, the cost has gone up really high. Okay, so in this case, I may go back to my scoring profile and say, well, now cost is too high. Please give me something that is cheaper, but still with good yield. So I push my cost up to high. High means high importance, okay? Not high cost, high importance. And then I can hit apply again. And then it will again look through the alternatives and see if it can find something better. So sometimes it doesn't come out with something better, right? Sometimes it will still show you the same thing. So it really depends on the options available. So this is a nice, I guess, a compromise. So it has a slightly lower yield than before. Previously it was 41%, but the cost was like $40,000. So this you compromise a little bit on the yield, but it's a lot. This route is a lot cheaper than before. Okay, so this is, you know, after playing around with this. So you can give this a try, but like I say, sometimes even after playing around, you don't always see a different route. Then in those situations, you have to go into each step manually to look at the options. All right, so this is the next thing we're going to look at under steps. So you can see here the steps as I move down the row, the column, different stages of the synthesis is highlighted. So it's describing the evidence and the alternatives available at each stage. Now you notice the second stage, right? B to C. I have essentially 0 alternatives. So this is the one and only option in SciFinder-N. But remember, this is experimental options because we have the predicted results off. So I would turn it on now. And if we go back into the steps, you can see now it has now 11 alternatives. Okay, so predicted results. So even for steps or compounds that already have experimental pathways, we would also give you predicted alternatives. So let's take a look at some of the alternatives. So I can go into, let's just go into the first stage here. Okay, let's assume we are not, you know, we don't like this approach. We go into the alternatives. Okay, this is the one that is currently selected. We can consider this, which was one of the other options we saw earlier on, or we can scroll down and look at other possibilities. Okay, so this is once it's in green, it's predicted. All right, and you can expand on the similar alternatives because we've kind of grouped pathways that are similar, like making the same bond formation together, so you don't see repetitive alternatives occurring. All right, so let's assume, okay, let's instead of doing the bromination, we're going to add in the phenol as the last step, right? And then there are various ways to get to this. As you can see here, it's all giving us the phenol. So we can select this instead. And what it does is it will input the new option that we have selected. Okay, now you can see this is in green because it's a predictive step. And this gives us new intermediates. So the subsequent pathway changes, we end up with a new starting material, and obviously the cost and the yield changes. Okay, so you can basically go into each stage, look at the alternatives, make the changes. It will re-update the route, recalculate the cost and price. All right. Now, the next thing you may ask is, if it's a predicted step, how do I know what reaction conditions to even use, right? So if it's experimental, it's very clear cut. If you click on evidence, right, for an experimental, let me just show you, it will be the actual exact reactions reported in the literature because it's experimental. So it has been done before, okay? So this is exactly what we see here. So when it's a predictive step, there is also evidence, okay? But the evidence won't be the exact starting materials that we see here. Just close this, right? Converting this to this, this to this. As you can see, it's not the same, not the exact same reactants, right? But it's the same type of chemistry. Okay, so even if I scroll down, it's like converting this aldehyde to the phenol, aldehyde to the phenol, but it is obviously a different starting material from this and this. Okay, so even for the predicted steps, we find the evidence, but we try to find as closely, you can see it's sorted by relevance, right? So it's structurally, we try to find as close as possible to what is in our retrosynthesis plan. Okay, so this is for predictive steps. Then the other thing you can do, all right, let me go to my plan options. So in this box over here, this is where you can specify the first bond to break in the retrosynthesis. So people always ask whether can they indicate their starting material, right? They say, I want to start with starting material A, and I want to end up with final compound D, okay? Just tell me how to do the in-between steps. So at the moment, you can't input starting material. Like I say, it's always just the target molecule. But what you can do is to use the break bond and protect bond function to try and influence the type of disconnection you see. So for example, if your starting material already has a bromine in place and you don't want to do any bromination reaction, you can say protect this bond. Okay, so for all the disconnections that you see, it won't try to break up this bond. You will just leave it as it is. And then you can also indicate exactly where you want the bond to be broken, the first bond to be broken, whether it's here, here, or here. Okay, so you can protect multiple bonds, as you can see here. But in terms of breaking bond, you can only indicate one. Right? Once I've selected one, I can't indicate another one. Okay, so let me redo this. Let's break this bond, protect this bond. Okay, so if I rerun the retrosynthesis plan, the new plan would fit this criteria that I have just indicated. So I think it will again take some time. Okay, and in this case, it doesn't find any experimental results for the new criteria that I have indicated. So let me go to the search history. Okay, it's also complete. So let's open this up. Okay, so you can see here, because we have indicated that the bromine bond cannot be broken, so it keeps it all the way. It tries its best to keep it all the way to the end. And then the first disconnection that we see is at this stage, at this bond here. Okay, so this is what you can do to influence the type of plans that you see. The other thing that you can also do, let me just maybe pull out this. Is this two other same? Okay, never mind. I'll use this. You can exclude certain steps or intermediate. So for example, for some reason, I don't like this step. I don't want to perform this type of chemistry. I can say exclude this step, and it will then try to find alternative way to get to your target molecule without going through that disconnection. Okay. All right, so you can see it's suggesting something else. And you can also say, I want to certain type of reagent or intermediate or reactant that you don't want to use. You can also exclude it. So there's also a trash bin here. So I'm just going to pick this one. And then it will, again, try to find an alternative for you. Okay, so I think it was something else here. And basically whatever we have excluded is sent to this box, so you can always recover it if you change your mind. Okay. So this is the retrosynthesis plan. I think it's very useful to help you plan synthesis. So you still have to, I guess, use your chemistry knowledge to decide, which pathway is more suitable for your situation. But now you have all the options pulled together onto one interface. You have the evidence pulled in for you. It calculates the yield and price, so you don't have to recalculate each time you make a change in your plan, right? So give it a try. And like I said, it works both for known molecules as well as unknown molecules, novel molecules, okay? Okay, so that's all I have for my demonstration. Like I say, there's still biosequence searching, but since most people are from chemistry, so I won't talk about this today, but let the library know if there's interest to learn about biosequence searching. And we have a couple of minutes for any questions you may have.
มีใครมีคำถามไหมคะ สามารถสอบถามได้เลยนะคะ เป็นภาษาไทยก็ได้ค่ะ มีไหมคะ ถ้าไม่มีเดี๋ยวขอถามเพิ่มเติมก็ได้ค่ะ ในส่วนของ reference นะคะที่ทางคุณแพมแจ้งว่าเรามีเอกสารฉบับเต็มแล้วก็ Open Access ถูกไหมคะอาจารย์ทีเนี้ยในส่วนที่มันเป็น Full Text นะคะคือตอนแรกคิดว่าในส่วนของฐานข้อมูลมันมี Full Text ในตัวเองด้วยแต่เมื่อกี้คุณแพมแจ้งว่าFull Text นั้นน่ะถ้าห้องสมุดไม่ได้มีสิทธิ์บอกรับหรือเขาเรียกนั้นไม่มีสิทธิ์เข้าใช้ไม่ได้บอกรับอย่างเงี้ยก็ดูไม่ได้เหรอคะหรือยังไง
ใช่ครับคิดว่าขึ้นอยู่กับว่าห้องสมุดเราได้ subscribe ไปด้วยหรือเปล่าครับ
งั้นก็แสดงว่าเราดูได้เฉพาะ 100% ในส่วนของ Open Access หรือเปล่าคะ
ใช่ครับแต่ว่าในส่วนของตัวที่มันไม่ได้ access full text ก็คือต้องดูเป็นพวก abstract หรืออะไรเงี้ยครับเป็น DOI ไปครับผม
ท่านอื่นมีคำถามไหมคะ
นิสิตมีคำถามไหมครับหรืออาจารย์ท่านอื่นก็ได้ครับผม
คือฐานก็อาจจะค่อนข้างซับซ้อนในส่วนของบรรณารักษ์ก็จะคุ้นเคยกับฐานข้อมูลที่เป็นe-book หรือว่าเป็น article อะไรพวกนี้ค่ะทีนี้ฐานนี้มันก็จะมีความเฉพาะทางอยู่สร้างสูตรอะไรอย่างเงี้ย
ใช่ครับแต่คิดว่านิสิต grad student ส่วนใหญ่น่าจะคุ้นเคยกันอยู่แล้วครับผมSo I don't think we have a couple like more questions so okay should we
ไม่มีเหรอคะ
ครับคิดว่าไม่น่าอาจารย์ท่านอื่นมีคำถามไหมครับ Is there any question from faculties or any grad students
เมื่อกี้จะมีคำถามหลังไมค์คือแชทแบบ direct มาที่ห้องสมุดนะคะสอบถามในเรื่องของการ ลงทะเบียนนะคะเดี๋ยวจะขออนุญาตแชร์หน้าจอนิดนึงนะคะอ๋อเดี๋ยวนะ
I can give everyone my email address. So if you
yes
have,
Okay.
yeah, if you have any questions later on, or when you're trying your own searches, feel free to reach out to me.
Thank you, Pam.
No problem.
สำหรับการลงทะเบียนการเข้าใช้ฐานข้อมูลนะคะสำหรับฐานนี้นะคะขอแจ้งอีกครั้งหนึ่งว่าฐาน SciFinder จะต้องมีการลงทะเบียนเพื่อให้มี Account ก่อนสำหรับคนเข้าใช้ครั้งแรกนะคะแล้วก็หลังจากนั้นถึงจะสามารถเข้าใช้ฐานได้โดยที่เราจะใช้ Account นั้นในการเข้าใช้งานนะคะจะต้องใช้งานผ่านOpenA10 เท่านั้นนะคะทีนี้ทุกท่านสามารถมาได้ที่เมนูนี้ก็ได้ค่ะเดี๋ยวให้น้องศิวพรส่งลิงก์ให้ทางแชทนะคะเป็นลิงก์ของเมนู Database แล้วก็มาที่ฐานข้อมูลทดลองใช้นะคะตอนนี้ฐานนี้เป็นฐานข้อมูลทดลองใช้อยู่นะคะพอมาถึงจะเจอฐาน SciFinder-N นะคะคลิกที่ตัว I จะมีรายละเอียดทั้งหมดนะคะว่าเป็นฐานเกี่ยวกับอะไรวันนี้ทางคุณแพมได้แนะนำนะคะว่าเราสามารถเข้าใช้หรือสืบค้นในส่วนของสสารหรือsubstance ได้นะคะหรือว่าเป็นปฏิกิริยาเคมีหรือว่าในส่วนของ reference นั้นก็จะเป็นการสืบค้นเป็นบทความนะคะซึ่งเราก็จะดูได้ในส่วนที่ Open Access หรือในส่วนที่เป็นบทความหรือฐานข้อมูลที่สำนักหอสมุดกลางมีสิทธิ์เข้าใช้นะคะแล้วก็ตรงนี้นะคะจะเป็นวิธีการใช้นะคะใครที่ยังไม่เคยลงทะเบียนนะคะครั้งแรกให้มาลงทะเบียนที่ลิงก์นี้นะคะให้เข้ามาทำที่ลิงก์ที่หน้าเว็บไซต์ห้องสมุดนะคะเนื่องจากเราจะทำการดำเนินการจัดการลิงก์ไว้ให้เรียบร้อยสำหรับการแสดงตัวตนผ่าน OpenA10 นะคะตรงนี้เราจะมีคู่มือการลงทะเบียนและการใช้งานไว้ให้เรียบร้อยนะคะในการลงทะเบียนนะคะมาคลิกลิงก์ลงทะเบียนใช้งานที่นี่นะคะหลังจากที่เราลงทะเบียนเรียบร้อยแล้วในการลงทะเบียนนะคะถ้าผู้ใช้ยังไม่ได้แสดงตัวตนผ่าน OpenA10 นะคะจะขึ้นหน้าต่างนี้ให้ก่อนนะคะเราก็ใส่บัวสี ID เข้าไปนะคะหลังจากนั้นเมื่อเราลงทะเบียนเรียบร้อยแล้วเวลาลงทะเบียนนะคะขออนุญาตให้ทุกท่านใช้เป็นอีเมลของ GSWU หรือ MSWU นะคะและพอเราลงทะเบียนเรียบร้อยแล้วทุกท่านจะได้รับอันนี้เป็นหน้าลงทะเบียนนะคะ next พอลงทะเบียนเรียบร้อยเนี่ยจะได้รับอีเมลนะคะจากระบบนะคะให้เราทำการยืนยันการลงทะเบียนอีกครั้งหนึ่งนะคะก็คลิกยืนยันไปจากอีเมลนะคะแล้วก็ go to SciFinder-N นะคะจะคลิกลิงก์ไหนก็ได้แล้วก็มันจะเข้าสู่หน้าฐานเลยแล้วก็จะล็อกอินในชื่อเราเรียบร้อยเลยนะคะใช้งานเสร็จก็ล็อกเอาท์แต่ถ้าเมื่อไหร่ที่เราต้องการจะใช้งานใหม่อันนี้เรามี Account แล้วถูกไหมคะก็สามารถมาคลิกลิงก์ใช้งานฐานได้ที่ตรงนี้นะคะลิงก์มันจะยาวหน่อยนะคะไม่ใช่ลิงก์ทั่วไปของฐาน SciFinder ก็ถ้าทุกท่านไม่อยากจะเข้ามาที่หน้าเว็บห้องสมุดก็ทำเป็นบุ๊กมาร์กไว้ก็ได้นะคะหลังจากนั้นแล้วเนี่ยถ้ายังไม่ได้แสดงตัวตนก็จะให้ใส่บัวสีเช่นเดียวกันนะคะแล้วก็จะให้ล็อกอินนะคะสามารถใส่เป็นusername หรือใส่เป็นอีเมลก็ได้นะคะที่เราลงทะเบียนไว้แล้วก็ next แต่ถ้าท่านใดที่อยู่นอกเครือข่ายอะค่ะมันจะขึ้นไปเลยว่ามันไม่มีสิทธิ์ในการเข้าใช้นะคะไม่ได้อยู่ใน IP ที่กำหนดนะคะแล้วก็ใส่รหัสเข้าไปนะคะก็จะสามารถเข้าใช้งานฐานข้อมูลได้นะคะแล้วก็จะมี Account เราโชว์อยู่ตรงนี้นะคะเนี่ยนะคะลองมาดูได้นะคะที่เมนู Database นะคะฐานข้อมูลทดลองใช้นะคะแล้วก็คลิกที่ตัว I เราจะใช้ฐานข้อมูลนี้ได้ถึงวันที่31ธันวาคมนะคะสำหรับฐานข้อมูลอื่นๆนะคะทุกท่านสามารถมาคลิกที่ฐานข้อมูลออนไลน์นะคะแยกตามคณะนะคะแล้วก็เข้าใช้งานได้เลยถ้าท่านใดยังไม่ได้แสดงตัวตนนะคะเมื่อไหร่ที่คลิกที่ชื่อฐานก็จะมีบัวสีให้ใส่แต่ถ้าไม่มีบัวสีให้ใส่นั่นก็แสดงว่าเราได้แสดงตัวตนไปแล้วนะคะการแสดงตัวตน1 ครั้งสามารถใช้งานได้8 ชั่วโมงนะคะแล้วก็สำหรับคลิปในวันนี้นะคะเราก็จะถ้าหลังจากที่เราดำเนินการเรียบร้อยแล้วนะคะเราก็จะนำขึ้นนะคะมาไว้ให้ที่ตัว I เช่นเดียวกันนะคะตรงนี้นะคะของฐาน SciFinder นะคะเราจะเอาคลิปมาแปะไว้ให้ตรงนี้นะคะแล้วก็อีกส่วนหนึ่งก็คือสามารถมาดูได้ที่หน้าเว็บไซต์ของสำนักหอสมุดกลางนะคะที่เมนู User Guide นะคะตรงนี้เราจะมีรวมคลิป นะคะในการใช้เครื่องมือสืบค้น Substitute Discovery Untitled N นะคะแล้วก็คลิปการอบรมฐานข้อมูลต่างๆจะอยู่ที่ Retrosynthesis นะคะจะมีการแนะนำวิธีการเชื่อมต่อ Open แล้วก็อันนี้ค่ะจะเป็นรวบรวมคลิปการอบรมหรือบรรยายโดยสำนักพิมพ์แล้วก็สำนักหอสมุดกลางนะคะสามารถเข้ามาดูได้ตรงนี้นะคะแล้วก็ท่านใดที่ลงทะเบียนนะคะทางห้องสมุดจะส่งลิงก์คลิปไปให้อีกทางหนึ่งด้วยนะคะแล้วก็ในช่วงท้ายนี้นะคะก็จะรบกวนทุกท่านประเมินการเข้าอบรมนะคะจากลิงก์ที่เดี๋ยวคุณศิวพรจะส่งให้ทางแชทนะคะแล้วก็เดี๋ยวจะส่งเพิ่มเติมให้ทางอีเมลด้วยนะคะในการติดต่อสอบถามวันนี้หากมีปัญหานะคะก็สามารถแจ้งมาได้ที่ติดต่อเราด้านล่างนี้นะคะ arclibrarian@gsabu นะคะอีเมลนะคะแล้วก็กดไลค์ Facebook นะคะเพื่อติดต่อกดไลค์ติดตามนะคะ Facebook Line นะคะเพื่อที่จะติดต่อสอบถามต่างๆได้นะคะเพราะเราจะมีกิจกรรมแล้วก็จัดอบรมอยู่เรื่อยๆโดยเฉพาะเดี๋ยวเราจะเข้ารอบปีงบประมาณใหม่นะคะจะมีฐานข้อมูลใหม่ๆมีการจัดอบรมหัวข้อใหม่ๆในช่วงต้นปีนะคะอยากให้ทุกท่านมาติดตามนะคะจะได้เข้าร่วมการอบรมกับเราได้นะคะตรงนี้ใครมีคำถามเพิ่มเติมไหมคะในการใช้งานมีไหมคะ
คิดว่าไม่น่ามีแล้วครับผม
โอเคนะคะถ้าไม่มีแล้วนะคะก็ในนามของสำนักหอสมุดกลางนะคะก็ขอขอบพระคุณนะคะอาจารย์อิทธิพลนะคะสังเวียนวงศ์นะคะที่จากภาควิชาเคมีคณะวิทยาศาสตร์นะคะที่กรุณามาช่วยในเรื่องของการแนะนำวิทยากรแล้วก็มาเตรียมในการที่จะช่วยแปลให้กับเรานะคะแล้วก็ in behalf of the Central Library of Sinakilin Willows University I would like to thank you Pamela Un from CAS for demonstrating how to use SciFinder-N to us today. I think this will help our user to be able to understand and use the database more effectively. Thank you very much.
It's my pleasure. Thank you. Thank you for organizing
Thank
this.
you.
Thank you.
Thank
แล้วก็
you.
สุดท้ายนะคะขอขอบคุณผู้เข้าร่วมอบรมทุกท่านเลยนะคะแล้วก็หวังว่าในการอบรมครั้งต่อๆไปนะคะก็จะทุกท่านก็จะมาเข้าร่วมกับเราอีกซึ่งก็เดี๋ยวขอให้ติดตามนะคะจากหน้า Facebook แล้วก็เว็บไซต์ของสำนักหอสมุดกลางนะคะวันนี้อัมพรขอลาไปก่อนแล้วนะคะพบกันโอกาสหน้าสวัสดีค่ะ
สวัสดีครับ
Thank you very
Thank
much.
you
สวัสดีครับขอบคุณมาก Thank you
Thank you everyone.
Thank you.
Have a good day.
You
Bye
too.
bye.